This paper is the review of Dynamics, Gene Transfer and Ecological Function of Intracellular and Extracellular DNA in Environmental Microbiome. The first author, Dr. Mao Yi, and co-first author, Dr. Zhanyun Zhang, come from the Institute of Soil Science, Chinese Academy of Sciences, China. The third author, Dr. Mingming Sun, comes from College of Resources and Environmental Sciences, Nanjing Agricultural University, China. And the corresponding author, Dr. Yu Shi, comes from School of Life Sciences, Henan University, China. This review mainly focuses on the distribution and environmental behavior of eDNAs, iDNAs in environmental microbiome, the role of eDNAs in biofilm formation, effective eDNAs, iDNAs in the gene transmission, thereby reviewing the difference between two DNA fractions in the environmental behavior and the genetic information harbored. eDNAs, iDNAs. DNA can be further divided into extracellular DNA and intracellular DNA. Therein, eDNA is cell-free, naked DNA that usually persists in both aquatic and terrestrial environments. The DNA that exists in the living cell is the so-called intracellular DNA, mainly being released by living cells or the lying of dead cells, for example, autophagocytosis, virus infection, and active transport. Extracellular DNA extensively presents in soil, aquifer, and sediment environments, and can be classified into free-living, loosely bound, and tightly bound eDNAs fractions. iDNAs exist within living cells, thus avoiding direct interaction with environmental organic matters, and they mainly involve the nutrient cycling and metabolism, lateral and horizontal gene transfer. Environmental dynamics. The behavior of eDNAs in various environments has focused on the absorption kinetics, degradation pathways, and gene transfer, while iDNA's behavior was closely correlated with predation by animals and microorganisms, and the transformation of iDNAs into eDNAs. Environmental properties including mineral composition, pH and humus, and cation content exerted cross-impact on the absorption kinetic of eDNAs in environmental microbiome. Hydrolysis, oxidation, chemical cross-linking, and exogenic nuclease degradation are all important processes that involve with the degradation of eDNAs in environment. Moreover, natural transformation of eDNAs is the main mechanism of eukaryotic absorption, incorporation, and expression of eDNAs. Biofilm formation. eDNAs, along with polysaccharide, fatty acid, protein, and nucleic acid, are organic components of biofilm. eDNAs not only introduces a favorable acid-base interaction, but also provides available thermodynamic conditions for bacterial aggression and adhesion to the surface. eDNAs can protect Pseudomonas aeruginosa from the impact of pollutants by chelating cations, for example, aminoglycosides, antibacterial peptides, and inducing antibiotic resistance in the biofilms. In addition, the chelation between eDNAs and divalent cation in S. epidermis biofilm increase the resistance of the biofilm to antibiotics. Biological function and gene transfer. Nuclease released by soil bacteria degraded eDNAs and generated nutrient elements that were available for uptake of bacteria and plants. Moreover, as figured by high-throughput sequencing, both iDNAs and eDNAs were consisted of abundant eukaryotic and prokaryotic information. Take antibiotic-resistant genes, for example. Comparing to free-living eDNAs, the absorbed eDNAs are more prone to combine with a recipient competent cell. Alongside with the current focus on research of iDNAs, it is the proper time to bring eDNAs into the light and investigate its contribution in the important matter of cycling and genetic information transmission process. Overall, this review sheds light on the difference and interaction in ecological functioning between eDNAs and iDNAs, therein stimulating the further exploring of corresponding mechanism between the two DNA fractions. This work was financially supported by the National Natural Science Foundation of China, the Youth Innovation of the Chinese Academy of Sciences, and National Key Research and Development Program of China.